पीसीआर प्रिंसिपल्स एंड प्रोसीजर्स। जैसा कि हमने डिस्कस किया कि पीसीआर को यूज़ किया जा सकता है टू एम्पलीफाई स्पेसिफिक डीएनए फ्रैगमेंट कि हमारे पास मिलियन ऑफ कॉपीज़ जो हैं किसी भी टारगेट सीक्वेंस की वो जनरेट हो सकती हैं बाय यूजिंग दिस टेक्नोलॉजी। Now, the target sequence that we may have focused, it may have different sizes or fragment lengths. For example, it may range from a few kilobases to several kilobases, like it may be up to 40 kilobases. And finally, the product that is accumulated, it may also be variable because it can be dependent upon the different factors of the reaction mixture, like what may be the concentration of the template DNA, and especially with reference to dark DNA polymerases, concentration of DNTPs, and temperature that we have used during the different steps of the PCR. So the yield, it may be affected. Now, with reference to reaction mixture where different components and reagents they have been incorporated. Now we can briefly focus that what may be different reagents that we can use during the process of DNA amplification. जैसे मैंने लास्ट टॉपिक में भी एक्सप्लेन किया कि जो बेसिक रिक्वायरमेंट है उनमें से डीएनए टेम्पलेट है कि हमें एक टारगेट डीएनए की ज़रूरत होती है जिसको हम एम्पलीफाई करना चाहते हैं, और इसमें जो टेम्पलेट की कॉन्सेंट्रेशन है अगेन इट मे बी वेरिएबल, like it may be 0.1 to 1 microgram, and that can be used in the reaction mixture. Then the next important requirement, it is the availability of the primer, ताकि जो हमने DNA sequence करना है ये उसको फ्लैंक करे, उसको सराउंड करे और फिर उसकी 5 प्राइम टू 3 प्राइम जो DNA synthesis है उसको ये डायरेक्ट करे अकॉर्डिंग टू द इंफॉर्मेशन दैट इज कंटेंट इन द टेम्पलेट और द टारगेट डीएनए। तो इसमें जो प्राइमर की कॉन्सेंट्रेशन है अगेन इट मे बी वेरिएबल डिपेंडिंग हाउ मच रिएक्शन मिक्सचर वी आर गोइंग टू यूज, सपोज इट मे बी 10 or 20 picomole. Next, it is the requirement of enzymes like DNA polymerases that can bind with DNA to DNA hybrid, that is the hybrid that is formed by the template DNA and the primer, and then it can move along the DNA and adds the corresponding bases according to the information that is contained in the template DNA. In addition to that, the DNTPs, that is nucleoside triphosphates, deoxynucleotide triphosphates, you can also say that, and in short we can say that different type of DNTPs that are mainly 4 in case of DNA, and they are also added in a particular concentration like maybe 20 micromole in case of this reaction mixture. And we also required buffered solution that provides stability to the TARC DNA polymerases, and of course some cations in the form of magnesium ions or manganese ion that are the form of divalent cations, and some monovalent cations like the potassium ion, they are also required in the reaction mixture. Now we have the reaction mixture in Eppendorf tubes that may have the size of around 0.2 to 0.5 ml, and in this tube the reaction mixture we can manage in different volume. It depends on our own requirement कि हम कितना वॉल्यूम जो है वो एम्पलीफाई करना चाह रहे हैं. लाइक इसमें हम 10 माइक्रोलीटर का भी रिएक्शन मिक्सचर यूज़ कर सकते हैं, 25, 50, 100, 200. It depends that how much amplified DNA we have the requirement. जब हमारा रिएक्शन मिक्सचर रेडी होता है तो फिर हम इसको थर्मल साइक्लर में जो है वो प्लेस करते हैं, थर्मल साइक्लर विच इज़ द PCR मशीन. तो उसमें हम इसको इंक्यूबेट करते हैं, एंड आफ्टर placing this in thermal cycler, the reaction mixture, it is subjected to a different type of temperatures. Mainly three main temperatures they are used that and now we are going to discuss one by one. So one of the initial step in PCR, it is the initialization step where the reaction mixture it is heated at very high temperature, like it may be up to 96 or 98 degrees Celsius, ताकि इसमें जो DNA जो polymerases हैं वो activate हो जाएं, and even it can also cause the denaturation of the DNA. इसके बाद जो फर्स्ट रेगुलर साइकिल है वो है डीनेचुरेशन स्टेप का. तो डीनेचुरेशन जो है हम इसको करते हैं एट हाई टेम्परेचर अगेन, लाइक मोर देन 90 डिग्री सेल्सियस, मे बी 94 डिग्री सेल्सियस फॉर ए फ्यू सेकंड्स, लाइक इट मे बी 20 to 30 seconds heat exposure to the reaction mixture, so that the template DNA that is present, it is denatured. Denaturation means the hydrogen bonding that is present between the complementary base pairing, it is disrupted, and the double-stranded DNA, it is converted into single-stranded form. After its conversion into single-stranded form, the next major step in the PCR after denaturation, it is the annealing of the primer. And this annealing step, it is carried relatively at low temperature, like it may be between 50 to 65 degree Celsius. और इसमें हम ये ख्याल रखते हैं कि जो annealing temperature है वो कम से कम 5 डिग्री सेल्सियस लो होना चाहिए to the melting temperature of the primer. तो primer जो है वो अपने complementary sequence से bind करता है on the target DNA, and then with the help of DNA polymerase, it can cause the extension of the DNA in the next step. So after annealing of the primer to the target sequence, we can say that the next step, it is the extension step, where the DNA in the presence of DNA polymerases, DNTPs, buffer solution, it can cause the elongation of the DNA into 5' to 3' end, so that DNA, it is now extended. And next, so these three steps, like denaturation, then annealing of the primer, and next it is the extension. It is repeated many times. So DNA, it is subjected to repeated cycles of denaturation, annealing, and extension. So at the end of the cycle, we may have a huge quantity of the DNA. And the next step is to expose the reaction mixture to the final elongation step. That may be up to, for example, like 5 to 15 minutes. Why this step is required? It is required so that if some single-stranded DNA, they are present in the reaction mixture, they may have also been extended. And some, if there are some bases that have not been incorporated, so they are totally repaired. So this may be the advantage of the final extension that is carried out for 5 to 15 minutes. And when all the steps they are completed, the next step, it is the holding the reaction mixture at low temperature. Low temperature means that when the reaction it is completed, तो हमें इसकी स्टोरेज की ज़रूरत होती है. तो टेम्पोरेरी स्टोरेज के लिए हम जो एम्पलीफाइड प्रोडक्ट है उसको PCR के मशीन में ही या थर्मोसाइक्लर में ही हम उसको स्टोर कर सकते हैं एट लो टेम्परेचर, लाइक मे बी 4 degree Celsius. So when all the steps they are completed, as I explained in last topic, that at the end, due to the exponential increase in the DNA concentration, we may have millions of copies of the DNA.